This lecture will be covering the structure of genes and chromosomes, and this is sort of a high-level view of DNA. Next lecture we'll be going into a little bit more of the details, looking at actual DNA sequences and that sort of thing. So probably going a little bit quickly through this section, just because the details we're not focused so much on sequence level details. Okay, this is chapter 24 from our book. Some of these slides are from a different book, which was chapter 9, so that's kind of why the numbering's different here. What we'll be talking about is the organization of this information into chromosomes, how DNA supercoils, how it's able to pack in so tightly, and the actual structure of a chromosome. DNA is a very big molecule. If you look at, and we'll see some examples of this, but when you look at the size of DNA, like the length of a DNA molecule in a given cell type, it's really quite a bit longer than the cells that it goes into. So it has to be organized and compact very tightly to fit in these cells. And that's where we get this term of chromosome. And that's when DNA is coiled and tightly packed, usually with the help of proteins. We call this a chromosome. This figure from our book shows a virus that infects bacteria cells. It's called a bacteriophage, and this one's specifically bacteriophage T2. And that's shown here in the middle. The DNA has spilled out here. In this electron micrograph, we can see the DNA, and it's this what looks like a string. So you can see how long this molecule is compared to the size of the virus that it fits into. Okay. DNA has a secondary structure. We typically think of that as the B form of DNA. We've talked about that in nucleotides when we covered those. That would be the secondary structure. There's also tertiary and quaternary structures of DNA as well. Okay? And all of these structural organizations lead it to be very tightly packed. Okay? And that's because it has to be. This link here will show exactly how DNA fits into a virus. It's an ATP motor. It uses ATP and it kind of packs the DNA in very tightly. So if you're interested in that, you can click on that link. All these links will be in the description below. The table 24.1 from our book shows a number of different bacteriophages, and really don't need to know this specifically. What's interesting though is they give the size of the DNA, and the size, as you can see, ranges drastically, and then the length of the viral DNA. Okay. Then what's interesting though is to compare the length of that DNA to the dimension of the viral particle. So the long side of the viral particle shown here, you can see that they're much, much smaller than the length of the DNA. So the DNA has to be tightly compacted to be able to fit in these. Okay. Viral DNA is usually very small compared to other bacteria DNA or obviously human DNA. It can be single-stranded RNA or it can be DNA. It's often circular. That's kind of a theme we'll see in viruses and in bacteria. So bacteria DNA is organized into one chromosome per cell. Okay, it's circular DNA and it's duplex, meaning it's double-stranded. Okay. And in this, you have one copy of each gene. Right? Some organisms, you actually have multiple copies of each gene, as in eukaryotic cells. Okay. Eukaryotic cells, the real difference here, in eukaryotic cells, there can be varying numbers of chromosomes. The thing about eukaryotic cells that's really interesting is that they have introns and exons. Right? So in the gene encoding regions of the DNA, there are these sequences called introns, which stands for intravening sequences. And it's actually a sequence that gets expressed, but it gets cleaved out before the messenger RNA is translated into protein. So it's you can kind of think of it as just like a junk sequence almost, although it's not really junk, but just a sequence that's inside a gene that does not get turned into the protein that the gene is going to become. Okay. And if you look at just how much of the human genome encodes protein, it's only about 1.5%, which is a very small amount. Okay. If you include the introns in that, it goes up to 30%. So you can see that the amount of introns in the genome is actually quite high. It's higher than the number of exons, or the regions that are getting turned into protein. Okay. There's also, if you take that away, that still leaves about 70% of the genome, right? So what other things make up that? There are things called transposons that are actually movable sections of DNA. Simple sequence DNA that have very simple repeats, also can be called satellite DNA. And then you have the centromere and telomere regions. If we look at bacteria first, right, how do they store the DNA? Well, again, it's double-stranded DNA. It's circular. It's a single chromosome. It's bigger than the bacteriophage chromosomes. Okay, so it's bigger than the virus DNA. Any extra chromosomal DNA, sometimes that you can have some extra chromosomal DNA in a bacteria, and we call that plasmid. Those are smaller circles of DNA. And in the biology or biochemistry laboratory, we take advantage of this by actually introducing these small circles or plasmid DNA into bacteria cells to make, for instance, to make a protein, express a protein that we want to purify. That's how it's historically done. Almost all DNA is in a gene or a sequence that regulates a gene. Okay, so you don't have, in a bacteria, you don't have this same amount of DNA that is not for expressing a gene or regulating a gene like you do in eukaryotic cells. Right, and here we have a picture of a E. coli cell that has been, the DNA has been let out of it. And the arrows here are pointing to some of these small circles that you get, which would actually be plasmid DNA. And the genome of the bacteria cell itself, the chromosome, is this much longer piece that's kind of coiled in on itself. Right, eukaryotic chromosomes are obviously much larger than bacterial chromosomes. And the DNA length in a typical eukaryotic cell is about 2 meters. Okay, so that's about 6 feet long. And you can imagine if you have, one cell, it's very, very small, right? So fitting that much DNA into one cell is hard to do. We call it diploid because there are 2 copies of each chromosome in the eukaryotic cells, or at least most eukaryotic cells. Okay. It's linear, so it's not circular. And because of that, you need a way to kind of protect the linear pieces of DNA. Human cells are about 20 micrometers in diameter. There is a huge range of that depending on cell type. So you can kind of compare that 2 meters DNA inside a 20 micrometer diameter cell. So the number of chromosomes, size of genome really does not directly relate to the complexity of an organism. And that's because most of the DNA is not for encoding genes directly. And because of this big length of DNA and the small size of the cell, it has to be highly organized. Okay. And that organized storage partially controls how the genes are expressed. If we look at pictures, kind of a cartoon description of chromosomes, right? You usually see these, kind of rod-shaped drawings, and you have bands on them, and those bands are where the genes would be. The centromere is there kind of in this, not directly center, but somewhere in the middle, and that's where the two copies of the chromosome, which we would call sister chromatids, those kind of stick together there by the centromere. And another way of looking at it is like this, right, where the centromere is here. So they're sort of X-shaped in a way. And how they really would look, if you look under these and under a powerful microscope, are shown here. And you can actually have these kind of analyzed in a lab to make sure that you don't have any chromosomal deficiencies. Right. So how we term this is collinear information, and that's because you have two strands of DNA, one going 5' to 3', and the other strand, the complementary strand, going in the opposite direction. It would be 5' to 3'. Okay. Okay. That is then turned into messenger RNA through the process of transcription, and the messenger RNA is turned into protein through the process of translation. Okay, that, in a nutshell, is the central dogma of biochemistry, which we will be discussing in the next few lectures, at the next 3 lectures at the end of class, at the end of this semester. So neither the total length of DNA nor the number of chromosomes correlates with the complexity of an organism. For an example, dogs have 78 chromosomes. Amphibians have much more DNA than humans do. Plants have more genes than humans. So it's not a very strong correlation there. And that, again, is because in eukaryotic cells, most of the DNA is non-coding. The biological significance of this non-coding DNA, it's not really entirely clear. There's still lots of research going on in this. If you think about it, the genome, the entire human genome was only sequenced, fairly recently, something like 20 years ago-ish, maybe 25 now, but it was, that in itself took many years to do the first time. Now we can do it very rapidly, so we've learned a lot in that time, but still there's a lot of genes that really we don't know what they do. Or we know there's a protein, but we still don't really know where the gene is for that protein. So there's still work being done on this. So this non-coding sequences, the significance of those is still pretty unclear. We know that some DNA regions directly participate in the regulation of gene expression. Okay, those are things like promoters, termination signals, and we'll be talking about those in the upcoming lectures. Some DNA encodes for small regulatory RNA, and those still we don't always know what exactly their functions are. And some DNA may just be, what we would refer to as junk, and that's pieces of unwanted genes, remnants of viral infections, things that are just sort of left over from, a different time, but have been incorporated into the genome. And, maybe evolutionarily there's a benefit to having this junk DNA around. As I mentioned before, if we're looking at genes specifically, there are two parts to a gene. One is the intron, or intervening sequence, and these are sequence of nucleotides in a gene that are transcribed but cut out before the gene is translated. So they're turned into RNA, but before that RNA is turned into protein, these intron regions are cut out. Okay. And the exon, it would be the opposite of that. That's the segment of the gene that encodes a portion of the final product or protein of the gene. And if we look at this, it's kind of interesting how the number of introns in genes can vary widely. Okay, so it's not like some consistent recipe. It's almost sort of random in a way. The two examples here, we have two genes, and the exons, the part that would be turned into a protein product, are shown in red, and the introns are shown in gray. So in this gene, we have 7 introns, and that takes into account 85% of that sequence of the gene. So it's a vast majority of this gene. Okay. Here in hemoglobin beta subunit, there are only 2 introns, but again, it's a really large amount of the sequence. Okay, but it's not always like this. Histone proteins, which we'll talk about when we're talking about the structure of a chromosome, histones don't have any introns. Okay, and a very large protein like titin, which is a muscle protein, has 178 introns. So the number of introns and how much of the sequence they compose of a gene can vary drastically. It has been determined in the recently, fairly recently, that bacteria genomes also can have introns. Not something we're going to be discussing in this class specifically, but just for your information, they do also contain some introns. So if we look at the chromosome, the parts of a chromosome, and we want to show it linearly laid out here, the end caps and the center of it are these sort of special structural regions. Okay, the ends, what we refer to as telomeres or telomeres, depending on how you pronounce that. I like to say telomere, but I've definitely heard it's pronounced telomere. And the middle is the centromere. Okay. And then the gray regions here would be the unique sequences that you have, or where the genes are. You have a lot of the repeats and the multiple replication origins as well. Okay. Centromere is the attachment point for proteins that link the chromosome to the mitotic spindle. So when the cells are dividing, that's how you make sure that each cell gets a copy, right? And the telomere, just the ends of the chromosome. And these telomere sequences help in stabilization, or thought to. The DNA, unlike bacteria or viral DNA, the eukaryotic DNA is linear, so you have to protect the ends from unwinding, things like that, or breaking off. So that's where these telomere regions are helpful. We mentioned earlier this thing called transposon, and those are sequences that can move within a genome. So eukaryotic genomes are not really completely static. They have these transposons that can move around within the genome of a single cell. The ends of the transposons do contain these repeat regions, and they account for an estimated 50% of the human genome. So a pretty large amount of the genome are these transposable regions. Right, telomeres are the caps, the end of the eukaryotic chromosomes, and they're thought to form these special loop structures to keep the DNA from unraveling. They're added by this enzyme telomerase. In many tissues, telomeres are shortened after each round of replication, and thus the cellular DNA can age because of that, right? And it's thought that one of the reasons we age is this telomere shrinkage, and there's research going on to actually study this and maybe prevent some of the bad parts about aging by being able to keep the telomeres extended or longer. And the sequences that we typically see in the telomere, the 5' end repeats are listed below. 3' end would be complementary. The repeat sequence in humans is TTA-GGG, and then that is repeated, a certain number of times. So for mammals, that number is about 1,500 times usually, right? And then again, it can get shortened as the cell divides. Right, centromeres, those are the regions where the two sister chromatids come together, and where the spindle can attach so that the daughter chromosomes are held and split apart during mitosis. Okay, these are comprised of AT-rich sequences and repeated sequences of close to 130 base pairs. So in this picture, this would be the centromere region. And we don't really go too much into the details of the centromere, only that they're AT-rich regions. But if you want to remind yourself what meiosis and mitosis are, this figure is good for that. We won't really be getting too much into the details of this, though, in this class. Okay, so how is this DNA packed in? Well, we saw the viral DNA, and it's associated or may be associated with what are capsid proteins, the proteins that are actually comprising the viral particle, the outside shell of the virus. Prokaryotic DNA is associated with proteins in what's referred to as the nucleoid, right? Bacteria or prokaryotic cells don't have a true nucleus, but they do have what's referred to as a nucleoid, where the DNA kind of packs in on itself and makes a sort of a quasi-nucleus. And eukaryotic DNA is organized with proteins into a complex called chromatin. Okay, so this combination of chromosome and protein is known as chromatin. So if we look at a picture here, we have the prokaryotic cell shown here, bacteria cell. And again, no nucleus, but this folded up chromosome, folded up and probably stuck together with some proteins there, that forms what's known as the nucleoid region. So comparing the structure of DNA, the level of organization from a prokaryotic cell to a eukaryotic cell. All right, in a prokaryotic cell, all you have is this circle of DNA, and it just sort of gets kind of coiled and wrapped along in on itself. This figure, by the way, is from a different textbook. Okay. Eukaryotic cell has a much more levels to the organization. So if we look on a secondary structure level, that would be shown here. The DNA, and we see that it's in that alpha helix, right? That the B form of DNA, that gets twisted around these little orange balls here. Those are histones. Those are histone proteins. So it gets coiled around those. Okay, clusters of those then get kind of coiled around each other, and they form what is sort of like a helix or a coil, right? And those coils of the DNA and the histones can coil again around themselves, and then those coiled coils can be wrapped in sort of a bundle together. Okay, and that again goes stuffed inside the nucleus. So you can see there's multiple levels of organization here within a eukaryotic cell, and we'll talk a little bit more in detail about these. Okay. Right, the analogy here is kind of like an old school phone cord. And I don't know if any of you have actually ever seen one of these, so I don't know how good of an analogy this is nowadays. But an old phone cord, you'd have the phone, and then it would be like stuck to something on the wall where the keypad was, and you'd pick it up and you'd go talk on it, and if you'd be walking around on it talking, the coil would get kind of wrapped around itself. So the coiled cable also kind of twists and wraps around itself. In this region where you get these twists around, there's a lot of tension. That's known as a supercoil. And that's kind of a good analogy to DNA. Just another way to look at this, the figure from the book, we have the DNA alpha helix. The B form of DNA is what we refer to as a relaxed form of DNA. So this, if it was just circled around that B form, that alpha helix being circled around, that'd be termed relax. Supercoiled DNA is when you have this circle actually twisting on itself. Okay, so that's shown in this. Okay. So there's some ramifications to supercoiling in DNA, and those occur in replication and transcription. Okay, as you, we'll go into detail about these processes in future lectures, so don't worry about getting all the details right now. But when DNA is in this case being transcribed into RNA, right, it kind of gets pulled apart here by the polymerase, right, and then the polymerase works through, and it adds to the RNA chain, and it moves forward. But when it's doing that, what you can see, it has to kind of twist the DNA here, and the DNA in front gets what's referred to as overwound. Okay, so the twists in this DNA are getting tighter and tighter. And then as you go back, the DNA behind it is underwound. So if you don't have any way to remedy this, the polymerase would actually at some point just stop, because it can no longer deal with that tension of the overwound DNA in front of it. So there's particular machinery to deal with this, and those are topoisomerases, which we'll talk about shortly. A link to visualize supercoiling, again, I'll just keep that there in the description box. I think this one was about 2 minutes long. If you want to just get a visual idea of what supercoiling is. So closed circular DNA, again, in the B form, is termed relaxed. Okay, there's one turn per about 10.5 base pairs in that helix. Okay. Changes are going to be due to structural strain, supercoiling, usually due to underwinding, and that means there are fewer helical turns. Underwinding makes later separation of the strands easier. And if we look in this electron micrograph, right, this would be the total relaxed, and then we have varying degrees of supercoiling going on, increasing in towards the right. Okay, and you can see the fragment that's most supercoiled is shown here, that it's really wrapped tightly around itself. Right, so there's some terminology with supercoiling that we need to go through. And first is the linking number, or LK. LK of 1 would be, totally relaxed DNA. Okay, and here we have LK of 6, and that denotes that it's crossing, one strand is crossing over the other strand 6 times. Okay. This is not a very good picture in reality. It's not really how it looks. But again, this is more of a cartoon to kind of help you understand what the terminology is. All right. In reality, you're not going to be looking at a picture to count the linking number. Okay. What you'll do is use a calculation. So linking number is the total number of base pairs divided by the number of base pairs per turn. Okay, so i.e. linking number then is really just the number of turns you have. Right, in this example, we have if we have a linking number of 200, right, and we remove 2 turns from that, our linking number will then be 198. Right, so if we have a relaxed B form of DNA with 2100 base pairs in that plasmid, we have 10.5 base pairs per turn. You do the math there using that equation above, that linking number is 200. Right, the change in linking number is just going to be your current linking number minus the linking number you started with. So, right, in the second example here, C, we have 198. We started with 200, so our delta LK is minus 2. Linking number depends on the length of the plasmid, okay? Because you're using the total number of base pairs divided by the base pairs per turn. Right? If you remove 2 turns, that delta LK is going to be negative, as we saw before. So going from 200 to 198, we get a delta LK of minus 2. If we add 2 turns, that's what we would refer to as being a positive supercoil. Okay? That would be delta LK of plus 2. Okay, so removing turns, we get negative supercoils. If we add turns, we get positive supercoils. There's also another term called superhelical density, which is abbreviated as the Greek letter sigma. It's just a measure of supercoiling, and that is a delta LK number divided by our LK naught. Okay, so for the previous example, if we had negative 2 as a delta LK, we divide that by what we started with in the relaxed form, which was 200, and that would be negative 0.01. So that in a sense is 1%. We could see that that's 1% negative supercoil. In vivo, the numbers that you typically see for this are negative 0.05 to negative 0.07. So that's about 5 to 7% underwound. Okay. If DNA is overwound, then it would get a positive value, but in vivo, usually it's underwound, and that's because it's easier to unravel and replicate or transcribe. Changing the linking number results in a change in supercoiling, right? Not surprising. And we refer to two forms of DNA that have the same sequence, but they only differ in terms of linking number as topoisomers. Okay, so that term topoisomer means that we have DNA that's the same sequence, it just has different linking numbers in it, i.e. they're different super helical densities. Right, and topoisomers differ by linking number. They can differ, by a linking number of 1 or 10, whatever. They just have different linking numbers. So this example from the book is shown here. Right, this circle of DNA has a linking number of 25. This one now is underwound. It has a linking number of 23. We would refer to those as topoisomers. Right. If you want to get really in detail, the negative supercoil here has a right-handed supercoil, and the positive has a left-handed, but I'm not going to expect you to know that in this case. Right, you can actually look at the degree of supercoiling in DNA on a gel. Compact supercoiled DNA travels faster in the gel than does the more relaxed DNA, right? Because as you supercoil it, it gets smaller and smaller. The physical size of it kind of comes in compared to what it looks like when it's relaxed. So these could travel faster. Okay, and that's just shown here on this agarose gel. So we have, in the first lane we have relaxed DNA and then highly supercoiled DNA. And then if we treat it with what's known as topoisomerase, an enzyme that changes linking numbers, we can get, bands here at every linking number. And so these single bands shown here are differing by a linking number of just 1. So you can separate DNA out when it's a change in linking number of only 1, which is pretty crazy. Okay, underwinding, as we've seen, most DNA in eukaryotic cells is underwound. Okay, and it facilitates additional structural changes. Okay, so it helps maintain structure of cruciforms at palindrome sequences. So this would be a cruciform structure that's much easier to have when the DNA is underwound compared to when it's completely relaxed. Okay, so cruciforms don't really occur in relaxed DNA, but they do in underwound DNA. It can also help the formation of left-handed Z form of DNA, which we really won't discuss a whole lot in this class. We just kind of assume that all DNA is in the B form, but in reality there are some regions that are in a left-handed Z form. Okay. These supercoils are only maintained if DNA is closed circle or bound by proteins. So that's another important reason why the eukaryotic chromosomes use proteins and are bound with proteins. Right, and these topoisomerases, right, these are the enzymes that kind of alluded to, but they're enzymes that change the linking number of DNA. And why they're important? Well, in DNA replication or transcription, right, you're kind of going through the DNA like a zipper, unwinding it, and if you don't really deal with that extra strain you're putting on the DNA in front of you, as we mentioned before, that's going to get overwound and it's going to basically turn into a knot, and you're not going to be able to continue through. So the topoisomerase enzymes kind of sit at the front there of, in this case, this is DNA replication, which we'll be discussing next lecture, right? And you have this topoisomerase there that can actually help cleave the DNA and unwind it in front, so you don't get that knot that will ultimately stop your replication. Right, there are two major types of topoisomerases. Type 1 works by making a cut in one DNA strand, and what they can do is relax negatively supercoiled DNA, i.e. increase the linking number. Okay, and there are type 2 topoisomerases that make cuts in both DNA strands, and what they do is induce negative supercoils, which would be decreasing the LK numbers. Right? And these type 2 actually use ATP. So E. coli has 4 topoisomerases, and this might get a little bit confusing. They have 4 separate topoisomerases. Topo 1 and 3 are type 1, right? Meaning that they relax negatively supercoiled DNA. And topo 2 is called DNA gyrase, which we'll talk more about. And it's actually a type 2 topoisomerase. Okay. Eukaryotic type 2 topoisomerases can't induce negative supercoils. They can only relax positive or negative supercoils. So there's a little bit of a difference there between bacteria and eukaryotic topoisomerases, which probably won't really become important in this class. Okay. So if we look at bacteria type 1 topoisomerase, the actual mechanism of it, they use a tyrosine residue to do this, and you'll see that the phosphate group that's connecting the 3' of one base and the 5' of another base, that tyrosine, the alcohol group on the tyrosine, is going to do a nucleophilic attack there on the phosphate group. Okay, and that breaks it, that single strand. Okay, so this mechanism, pretty simple, right? You just have a nucleophilic attack there. You're breaking one strand of DNA. Okay, so if you're looking at the cartoon picture of this, right, the tyrosine here is going to be breaking this strand. Okay. And then what happens after it breaks that, the other strand can kind of be pulled through that hole. And by doing that, you're changing the linking number by +1. Okay, you're actually adding another turn or coil in there. Okay. After that is pulled through, then the reverse reaction can happen. You get a nucleophilic attack from the 3' hydroxyl to the phosphate, okay, and everything goes back. So very simple mechanism, and you're just changing the linking number, again, by +1. If we look at topoisomerase 2, bacterial, now this is, we also refer to this as DNA gyrase. Okay. These break both strands, pass the DNA segment through the break, and then you join the strands. By doing this, you're changing the linking number as before, but you're changing it in increments of 2. All right, and you need ATP for this. And this is a way where the bacteria cells can actually induce negative supercoils, which decreases the linking number. It doesn't really happen that way in eukaryotic cells. We'll mention those here in a bit. They do decrease the linking number, but it's the way they do it, it's a little bit differently. It involves the histone proteins. Okay. So eukaryotic type 2 topoisomerases relax positive or negative supercoils. Therefore, it can increase or decrease the linking number. Okay, and again, ATP is being used in this. All right, so the mechanism of type 2 topoisomerases. Okay, if we start here, we have 2 strands, double-stranded DNA. The topoisomerase is kind of wrapped around this lighter colored blue strand. Okay, the darker colored blue strand is just a region of DNA a little bit further away from it. Okay, that binds to Then we get this break in DNA, the double-stranded DNA, the light blue that's sort of surrounded there in the topoisomerase. Okay. When that happens, then ATP is used to pull the darker blue strand through the light blue strand, the cut in it. Right? Then we can actually fuse those regions together by using that ATP, right? And because we've pulled that through, we have actually changed that linking number. Okay, and again, tyrosine is the residue, the residue in the active site there. Right, if you want to visualize the functions of these topoisomerases, there are links here you can view. Right, there's actually inhibitors made for topoisomerases, right? Because if these topoisomerases don't function, cells can't undergo DNA replication and they can't divide. Okay, so that's pretty big, right? So what circumstances would you want this to happen? Well, if you have a bacterial infection, right? The bacteria cells are going to be dividing much faster than your cells. So if you can make a topoisomerase inhibitor specific to the bacteria cells, that would be a very good target for an antibiotic. Okay. And some of those are shown here. Okay. Again, you're not going to be responsible for these structures, but just know that this is a target for antibiotics. Okay. They can also be used as chemotherapy agents, right? Because cancer cells are sort of rapidly growing cells. So if you can slow their cell division down by inhibiting their topoisomerase and making the division, the replication of their genomes impossible, that'd be a good target for a chemotherapy drug. Okay, and some of those are shown here. So in summary, topoisomerases control linking number and thus the supercoiling in a segment of DNA. Okay, there are two types. Type 1 breaks one strand and that changes the linking number by increments of 1. Generally, it relaxes DNA by removing negative supercoils, which would increase the linking number. Type 2 break both strands and changes the linking number by increments of 2. Okay. In bacteria cells, they are introducing negative supercoils, which decreases the linking number. Okay, they use ATP, okay, and in bacteria cells, it's referred to as DNA gyrase. Eukaryotic type 2 topoisomerase, they can't underwind the DNA, they can't induce negative supercoils, but they can both relax positive and negative supercoils, and the mechanism is the same. Right, so because of this, topoisomerases give us a mechanism for coiling or uncoiling to make chromosomes. So again, chromatin is a material that makes up chromosomes. It's complex of DNA, histone proteins, and some other proteins. Right? And it's about equal mass DNA and protein, which might be kind of surprising, right? Because you think of a chromosome as being all DNA, but actually it's about equal protein to DNA if you're looking at the mass. Okay. So histones are proteins that associate with DNA and allow it to become tightly wound together. Okay. What we refer to as a nucleosome, that's a structural unit for packing chromatin. It consists of DNA strand around a histone core. So if you hear that term nucleosome, what that means is you have DNA wound around histone, a histone protein. Right, so if we look at a cartoon picture of this, our histone core of the nucleosome is shown here, and there are 4 different colored wedges. And that's because the histones are actually 2 sets of 4 proteins kind of bound together. So there are total 8 histone proteins in this core. Okay. The DNA wraps around it, and then you have these linker regions of DNA that go between one of these nucleosomes to another. And that about 146 base pairs of DNA are wrapped around a histone, just for your information. When you look at it under electron micrograph, it looks like this. Kind of looks like beads on a string. The string linker DNA regions between them is about 54 base pairs. The N-terminal tails of histones stick out to form sites for modification, and we'll talk about these a little bit. And they also are important between making contacts between the nucleosomes, but they aren't actually shown here in this picture. Okay, histones, the molecular weight, can vary a little bit, but about 11,000 to 21,000 Daltons. They're rich in basic amino acids, so things like arginine, lysine, and obviously histamine. That is where they get the name histones. Excuse me, histidine, not histamine. So that's these basic amino acids. And that's important because the basic amino acids have positive charges, which can then associate with the negative phosphate charge in the DNA. There are 5 major types. They differ in their molecular weight and amino acid composition. These histone proteins can be heavily modified, and those are referred to as post-translational modifications. So that occurs after the protein has been translated. And things like the adding of a methyl group, an ADP, a phosphate group being added, sugars being added to them, acetylation. So all of these things, you can modify the histones, and that modification actually influences DNA packing and gene regulation. Modifications do change with age, and this is sort of an area of what's referred to as epigenetics, and that's something, a very new field, and it's the study of how your genes can change with respect to time or environment, how the DNA code itself doesn't change, but the way the genes are expressed is what's changing. Okay. And we've made kind of allusions to this, how the topoisomerase number 2 in eukaryotic cells can't induce negative supercoils. How you actually get underwound DNA in a eukaryotic cell is depicted here. So a histone protein, the DNA wraps around it, and it wraps around the histone in a negative supercoil. Okay, when it does that, it actually induces a positive supercoil on that unwound part. So the unbound part, if you use a topoisomerase on it, right, to relax that section of positive supercoil, the net effect of that is changing the linking number by -1, and then thus you get this one net negative supercoil because you have a negative supercoil on the histone. Okay, so that's just, maybe it's kind of semantics, but you can kind of think of this is how you get negative supercoiling in a eukaryotic cell, even though topoisomerase can't in itself generate a negative supercoil. Right. Looking a little bit closer on this, the histone proteins, we have a tetramer, a dimer of a tetramer, if that makes sense. Okay, there are 8 separate histone proteins. I should say 4 separate histone proteins, and you have 2 copies of them. They're referred to here as H2A and H2B, H3 and H4. Okay, that makes up the tetramer, and then there's a dimer of it. So there on this side there's, and you can see that they sort of are flipped. H4 is matching up to the H3 on the next dimer, H3 matching up with H4, and etc. Okay. That's what the histone core, the protein makeup of the histone core looks like. Right? You might say, okay, there's H2, H3, and H4, right? The H standing for histone. There's no H1. Where's the H1? We'll see that in a second. A little bit more detailed or realistic view is shown here. Not going to need to know this. The only thing that this shows that's good is that you have these the sort of terminal region, the N-terminal regions. You see how they stick out. Those are the regions that can be modified to induce different binding of the DNA or different gene expression. Again, here's just again another more detailed view of a sort of crystallography view of it, with the space filling model shown here, and you can see this blue double helical DNA wrapping around that histone core. The DNA wraps about 1 2/3 times around it. Okay, again, there's 146 bases that wrap around that histone, and it's a left-handed supercoil. Histone protein H1 is actually not part of this nucleosome bead, but it sits on that bead, and you can kind of think of it as a way to maybe lock in that coil around that nucleosome. Okay, it's sort of, I guess, lock would be the term that I would use for it. It also binds the linker DNA region between the nucleosomes, and it helps stabilize a zigzagged 30 nanometer chromatin fiber, which we'll see in a second. Okay, so that's where the H1 protein is. Okay, it's not actually part of the nucleosome bead itself, but it helps stabilize it from the outside. Well, there's actually a specific DNA sequencing that helps bind to a nucleosome, okay, and that is AT pairs. So AT base pairs allow the DNA to bend, right? GC bases don't really allow this bending as much. So the spacing is, you have about 2 or more AT bases at about 10 base pair intervals, and that allows the DNA to make a circle. Okay, so some of these repeat regions we see in DNA are actually just regions that help facilitate binding to chromatin, binding to histone proteins to make the chromatin. Right, and the histone tails, as they're referred to, these are the N-terminal regions of these histone proteins, and as you can see, they do stick out quite a ways, right? And those are the regions that you get modifications to, right? And they can actually line up together to sort of facilitate a interaction between separate nucleosome beads. So we have, once we have our DNA wrapped around our histone proteins to make these nucleosome cores, they can have a helical structure around themselves. Okay? And that's referred to as the 30 nanometer fiber, and that's shown here in electron micrograph. Okay, that's nucleosome formation compacting DNA about 7-fold. Okay. there's about 100-fold compaction here in the 30 nanometer fiber. So the nucleosome, when we just wrap DNA around a histone protein, that's a 7-fold compaction. Okay. Overall, the compaction in DNA to get in a cell is about 10,000-fold. Okay. So this nucleosome DNA wrapping around histones is only 7-fold. When we get to the 30 nanometer fiber, that represents a 100-fold compaction. Okay, so you can still see there's more folding and compacting that needs to go on. Right, this is not over the entire chromosome. It correlates with transcriptional activity. Higher order structures are still not that well understood. Okay, so the 30 nanometer fiber, there's a couple different models of it, but really what you can think of it as just these nucleosome cores kind of making a helix around themselves. So remember this dark brown strand is DNA. It's in a helix and it's getting wrapped around these histone proteins to make nucleosomes. So that helix is also making a helix around itself. If we look at these different orders of structures, right, we have starting with DNA with the double helix. That DNA double helix is wrapping around histones to make nucleosomes. Okay, that if we look at the size of that, that's about 11 nanometers, right? If we have those wrapping and making a helix around themselves, right? And if we're numbering them here, the histones are numbered 1 through 10. They're numbered 1 through 10 here. Okay, this is what's the 30 nanometer fiber. And then the higher order structures are shown here as well, which again, you probably don't need to know these as much in this class because they are, as we've said, not as well understood. But you're just making like more coils, coiled coils, okay, in a sense. We can see that it is a very complex structure. All right, and here's a example of this. If we have our nucleosome structures here, those are just being twisted on themselves and making a secondary helical structure to make the 30 nanometer fiber. I like this figure too. I think this figure is probably one of the better ones at showing these levels of structure, right? Starting from the double helix, which is about 2 nanometers, to the nucleosome structure, which is about 11 nanometers, 30 nanometer fiber, which is nucleosomes coiled on themselves, and then that is coiled up again, right, on itself, which makes 300 nanometer. The 700 nanometer fiber is, that again, making another coil, and so forth. And then finally, when you have that chromatid structure there of the chromosome, that's the 1400 nanometer structure. A video to kind of view this DNA compaction and storage is shown here. Again, that's in the description. Well, these chromosome, the condensation of chromosome structure is maintained, right? You can think of, you get this DNA coiled upon itself like that. There's got to be some pretty decent potential energy in it, and if you did that, let it go, it might just spring out, right, and explode out. So there are proteins that kind of help this structure be maintained, hold it together, and they're referred to as SMC proteins, which stands for structural maintenance of chromosomes. Right. And they have a basic structure shown here, kind of cartoon picture, where you have a hinge region and then the N-terminal and C-terminal. Right? And these are coiled. These straight pieces here are referred to as coiled coils. So this would be a monomer. Okay, when it to be active, it links with another copy and makes a dimer. Okay. These are found in all organisms, from bacteria to humans. There are two members of these SMC proteins. cohesions and condensins. Okay, cohesions keep two sister chromatids together. Condensins facilitate condensation of chromosome during cell division. Right? They bind DNA to cause DNA to be overwound, or positive supercoils. And their role is still not completely understood. If we look at them, this would be condensin. Okay, it looks kind of like cherries, I guess, that dimer. Those ATP binding regions that we saw, those are the ones that interact and wrap DNA into positive supercoils. Okay, they'll induce these positive supercoils, right? And then by doing so, you get negative supercoils on this unbound side. That's where topoisomerase 1 comes in, that then cleaves those negative supercoils. And then you lose that condensin unbinds. And what you've done there, the net result is you've added positive supercoiling to the DNA. Okay, so that concludes our discussion on chromosomes and genes. What we'll be discussing next time is DNA structure and actually the process of DNA replication. And we'll be moving into the last 3 lectures discussing the central dogma of biochemistry, that is taking DNA and replicating it, or being able to move DNA information to RNA information and then into protein information.